Hello and welcome everyone to our webinar series, New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. Today's webinar's title is Experimental Methods for Identifying Synergistic Nanobody Combinations that Neutralize SARS-CoV-2 by Dr. Fragments. My name is Cindy Cook and I'm the project lead for the Bioprotocol webinar series. For those of you who aren't familiar with Bioprotocol, Bioprotocol is an open access online journal whose mission is to make research and science more transparent and reproducible. So thank you all for being here and for helping us making this vision happen. And now it is my great pleasure to introduce our moderator of today's session, Dr. Peter Friday. Peter Friday is a research associate in the Laboratory of Cellular and Structural Biology at the Rockefeller University, where he also received his PhD. Over the last 10 years, he has worked extensively on the generation and characterization of camelid nanobodies, including the development of a novel pipeline for nanobody identification based on mass spectrometry and high-throughput DNA sequencing. He's collaborated on the development of hundreds of nanobodies against targets ranging from commonly used tags to disease-related proteins, including antigens from numerous pathogens. In addition to serving as a research tool, many of these nanobodies are currently being pursued for immunotherapeutic and diagnostic applications. Peter, thank you very much for being here, and I'm handing it over to you now.
All right, thank you very much. So it's my pleasure to introduce our speaker today, Dr. Fred Mast, who is a longtime expert in system cell biology. He's developed a range of tools in this field. They've applied to a number of different research problems in both cell biology and infectious disease. In particular, he's worked extensively on peroxisome biogenesis and function, going all the way back to his PhD studies, exploring the dynamic processes of organelle formation and regulation and their role in disease. He's also worked on applying new systems-level methods to infectious diseases, ranging from parasites like Plasmodia to viruses including dengue and most recently SARS-CoV-2. He's approached these problems from several different angles, studying pathogen biology, host response, as well as developing new therapeutics and diagnostics. In addressing these various disease-related problems, he's helped establish new high-throughput experimental methods, along with the quantitative tools and models to understand them. Dr. Mast is currently a senior research scientist at the Center for Global Infectious Disease Research at Seattle Children's Research Institute. He earlier did his postdoctoral work in Seattle at the Institute for Systems Biology with John Aitchison, after his graduate work at the University of Alberta with Richard Rajczynski. And I've actually had the pleasure of collaborating quite closely with Fred over the last few years, particularly on today's topic, applying nanobodies to the neutralization of SARS-CoV-2 spike. So later I'll be giving a little bit of background on the nanobody technologies behind the applications that Fred will be discussing. But with that, I'll hand it over to Fred.
Thanks, Peter, for that kind introduction, and thank you, Cindy, for the invite here to present at the Bioprotocol webinars. It's a great series, and I've enjoyed looking at some of the previous presentations and hope to contribute to an interesting one today. So as mentioned, the focus of our webinar today is really going to be about an experimental approach or an ensemble of approaches that we've put together to characterize and study a bunch of nanobodies against the spike protein of SARS-CoV-2. And as Peter mentioned, he'll give a brief introduction to those nanobodies and the methods that he's developed for generating them. And if you have any questions, please feel free to put them in the chat, and our colleague Natalia Katarin will be happy to answer them as we go along. So before I begin, I just want to provide some acknowledgement. This has been a large team effort that I've been proud to be a part of, and much of the work I'm presenting today was done in collaboration with this group of my colleagues. So that's Natalia, Peter, Paul, Erica, Jinji, Tan Moy. And we work together with John, Brian, and Mike as part of the National Center for Dynamic Interactome Research. This is a NIH-funded center in the United States for interactome research. And when coronavirus kind of came and descended upon us globally, we gathered together and decided to apply some of the tools that we have here within the center to tackling this problem and generating these nanobodies. In addition to these primary colleagues and contributors, we've also been able to have a wealth of additional partnerships, and some of that work I'll also present as we go through the webinar today. And just a brief disclosure, we are inventors obviously on patents describing these nanobodies. So the hope for today is to provide you with an overview, an introduction to nanobodies for those of you who are not familiar with them, as well as our unique approaches to generating nanobodies against specific targets. And then following from that generation of a repertoire against a particular target, in this case, spike, the methods that we've taken to biochemically characterize these nanobodies and then study their neutralization properties using both pseudovirus assays and live virus assays, both as individual neutralizers and also then in combination to demonstrate a synergistic potential of these nanobodies. And so I'll hand it over to Peter now to just provide a brief introduction.
All right, thanks. So antibodies, of course, have enormous value in research medicine as very high specificity, high affinity reagents that could be generated against pretty much any molecule of interest. And many of you are probably familiar with this kind of diagram of the standard IgG antibody molecule. So these are fairly large proteins, about 150 kilodaltons, and they're made up of 4 polypeptide chains, 2 heavy chains, shown in blue here, and then 2 light chains in green. But the antigen binding activity is actually just at the very ends of these heavy and light chains. And so we're interested in a variant of this. So in contrast to this typical antibody, nanobodies are actually derived from this unusual variant from camelid species like llamas and alpacas that lacks light chains. So they still bind the antigens with just as high affinity as a standard antibody, but instead they're doing this purely on their heavy chains at the ends of these variable domains. And so nanobodies themselves are actually just cloned from these heavy chain only antibodies and can be expressed in bacteria or other systems easily. So they're about 1/10 the size of a full nanobody, but full antibody, but they can bind with essentially equivalent performance. And so this is done by a couple of unique features of nanobodies. So they have an unusually stable framework region that allows them to be highly stable and soluble. They also tend to have larger CDR regions that can allow them to bind a larger epitope and so retain very high affinity even at this in this much smaller package. And so there are many advantages to these nanobodies. So as small single domain proteins, they're very easy to clone and express, particularly when you compare these to a conventional monoclonal. They're also very stable and soluble, as I said, and their small size offers a number of additional advantages potentially. So they can sometimes have much better epitope access for certain antigens, as they can fit in spaces that a normal antibody can't, since they're so much smaller. You can also have the potential for multiple nanobodies to bind on a single antigen if they hit different epitopes, which is something that we'll get into later to be very advantageous. They're also extremely easy to engineer. They're coded just in synthetic genes, so those can be modified as needed. And you can also combine nanobodies, 2 or 3 or 4 copies to make oligomers as needed. And as you'll see later, we can also make very large repertoires of these from even a single immunized llama relatively quickly. Traditionally, nanobodies are made through phage display or yeast display methods where you pan a library against your antigen of interest. And those approaches still work great. People use them all the time to get good nanobodies. But there's some disadvantages to that approach. So we developed an alternative that avoids going through that display step and instead looks at nanobody sequences going directly from serum samples. So just quickly to give you an overview of this process, we immunize a llama with an antigen or a mix of antigens of interest. And then we can then take bone marrow samples from that, which are enriched in lymphocytes that are expressing the antibodies. We can then take cDNA from those and sequence all of the heavy chain antibodies that llama is producing. In parallel to that, we can also take crude serum sample from the same animal and isolate the heavy chain variants directly from that sera and affinity purify them against the antigen of interest, very high stringency to get very high affinity binders. We can then analyze that by mass spectrometry to get peptide sequences that we can then match to the full-length DNA sequence library. And from that, we can get a large number of nanobody candidates that we can then express in bacteria and validate and use in the applications that Fred will discuss next.
Thanks, Peter. So as I mentioned, at the start of the pandemic, we decided to apply this unique approach of ours against the spike protein of SARS-CoV-2. And spike is a large glycoprotein. It exists as a trimer on the virion surface. This is a ribbon diagram of that spike trimer itself. And it's composed of two subunits, the S1 subunit, which also contains the receptor binding domain of spike, as well as the S2 subunit. And the RBD receptor binding domain of spike is important for engaging with the host cell receptor ACE2, whereas the S2 subunit is responsible for actually the fusion step that fuses the virion membrane with the host cell membrane. So using recombinantly expressed S1 subunit, S2 subunit, and as well as the receptor binding domain, these were all immunized. Llamas were all immunized with these various constructs, and from that we generated a very large repertoire of nanobodies. What you can see in this slide on the left panel here is a clustergram of all of the various unique CDR3s. These are the epitope binding sites on those nanobodies. We in total had over 375, with some of them closely related to each other, as indicated here in these clusters, but actually quite a lot which were not related to each other at the sequence level. And that's both for the S1 subunit and the RBD, which is all of these guys out here. By and large, the most nanobodies were against these two antigens, but we also got quite a fair number against the S2 antigen as well, the S2 subunit. Peter then took these and went through a series of screening, both for expression and affinity binding to these antigens, as he's mentioned. And we selected from this 375, 116, which are represented here in this clustal dendrogram, which essentially shows the extensive diversity of the repertoire. So very few of them are related to each other, and this large sequence diversity then offers us an advantage in terms of moving forward with identifying unique characteristics and really what might be the best sorts of nanobodies to target spike. So these were then subjected to a series of biochemical and biophysical characterizations, both to understand their on and off rates, or the kinetics, how they interact with their antigen of interest. This is done by surface plasmon resonance, where we can measure the fraction of bound to unbound over time and derive from it then our various kinetics. And you can see here for those 116 that we selected, by and large, most of them are below nanomolar range affinities, some dipping down into the picomolar affinities. So very fast on rates and low KDs exhibited across the repertoire. Didn't matter which the antigen was, whether it was RBD, S1, or S2. And the other neat thing about these nanobodies is that when you then look at these sensograms a little bit more closely, you can see that for many of them, they have very slow off rates. So they're quickly binding, and they're not really going to come off. And this is an excellent property to develop putative neutralizing therapeutics from. Nat also did a bunch of further characterizations to demonstrate their stability. So you can see here across the repertoire, when we looked at their melting temperatures, that they're actually quite stable. It requires most of them to get over 60 degrees Celsius before we start to see any sorts of denaturation. And if you were to go through a series of freeze or post-freeze dry steps, both their affinities and their melting temperatures are consistent. So these nanobodies are extremely stable. You can freeze dry them. We regularly have them sitting in the fridge, and we can pull out an aliquot that was made a year or 2 years before, and it still retains all of its biochemical activity, ability to target spike, ability to neutralize spike And so this is a rather advantageous if you think about wanting to deploy a therapeutic globally and not have to worry about shipping or temperature concerns. So from all of that characterization, we then moved into doing what's commonly referred to as epitope binning. And the idea here is to try find out, are these nanobodies binding to the same site on spike, or are they binding to different sites? And this was done using biolayer interferometry. So what we do is we take our antigen, we coat a glass tip that's read out on this machine here with the antigen itself. In this case, this is a representation of the receptor binding domain. We then pre-incubate with one nanobody, which will then engage with its antigen. And then we look and see if a subsequent different nanobody is also able to bind at the same time, or if this nanobody blocks its binding ability. And that's read out here. So you can see various pairwise combinations of these nanobodies with respect to each other. And we get various response values out of the machine. And then using this sort of data, we can then cluster that and look for whether or not the binding pattern of one nanobody against all other nanobodies is similar or dissimilar. So this is reporting Pearson correlation coefficients from all of that epitope binning data across all of our RBD specific binders. And you can see here that they group then into various clusters. And so this red cluster here, this group of nanobodies are nanobodies that are able to bind and would not block necessarily anything else here that you see in blue, but would block each other. So they have very similar binding characteristics with respect to their antigen. And so they have probably very similar or closely aligned epitopes on the RBD. And so that was done for both the RBD and then also the S1. So regions outside of the RBD, so the non-RBD epitope bins is demonstrated here, and then also for our S2s. And you can see that we have quite a few different epitope bins across spike from this nanobody repertoire. So now that we have that information, we understand, that they're very well at binding their targets, that they're very stable, and that we can bin them into these various bins. The next question was, what do they do? Are they able to have any sort of functional influence on blocking the ability of spike to engage ACE2 and or the ability of spike to engage in a fusion step with the membrane? And this is a really nice cartoon that I pulled from a review by Chmielewska and colleagues to highlight the sorts of approaches that you can take then to characterize neutralization. And we've ended up using all of these assays, and I'll go through what we've been doing and the benefits of some of them over others. But essentially, you can take cells that are receptive to SARS-CoV-2. The very common cell line for this approach is VeroE6 cells. They're used quite extensively throughout virology in terms of they're quite permissible for virus infection. They don't really have a strong innate immune response, and so they allow most viruses to be cultured inside of them. Or you can take something like a HEC293 cell that's been engineered to overexpress the ACE2 receptor on its surface and or other cell lines. And then these then are a little more specific for SARS-CoV-2 itself, and they've got ACE2 in high abundance. And if you have a virion present, they'll engage. And so you can either take the wild type virus, the authentic virus itself. This requires a biosafety level 3 to conduct that work. So it's a little more laborious. Or you can take this spike protein and you can encode it in pseudotyped viral particles. And there's two flavors that are commonly used for this approach. One is to replace VSV's G protein with spike, such that now this virus no longer has its normal coat, but it actually has the spike coat. And if you package then in additionally a reporter, you can then add this to cells. This will get in and they will then express that reporter, and you know that they've been successful in infection. The other approach is to use a lentivirus packaging system. And this is here then where you take an express spike on the lentivirus particle, and again, replace its genome with a plasmid that's going to have a readout, a fluorescence or luciferase-based readout. The one that we ended up choosing as the predominant method for going forward with our work is the lentivirus approach. Specifically, we took our lead from a colleague of ours at the Fred Hutch, Jesse Bloom, who had developed this assay really quickly early on in the pandemic, and basically got this working up in-house and made some modifications to make it a little more high throughput in our hands. So as I mentioned, this system, you basically have plasmids for the lentivirus, for your spike protein, and you can use SARS-CoV-2 spike, you can use the original SARS spike, various variants of concern or point mutants as you generate, and we've definitely taken advantage of that. And then as well, you package in a genome, which is basically a luciferase with a zombie green, and so you can read this out using standard biochemical methods for microscopy as your needs require. We then take these pseudotyped lentiviral particles and we add them to HEC293 cells that are expressing ACE2. And so if there's no inhibition, these two will readily infect these cells and you'll see them light up. But in the case of our nanobodies present, now the question is whether they're able to block that step. Through this, we've set up an automated system that will basically start with higher concentrations of our nanobodies, prepare a serial dilution of them, and then add and incubate them with the same concentration of the pseudotyped lentiviral particles. After that incubation step, we add them to the HEC293 cells, and after a brief incubation to allow luciferase to be produced, we can then add a substrate and read this out on a luminometer, and from that extract neutralization curves that are in a dose-dependent way tell us whether the nanobody is neutralizing or not neutralizing. This is just some high-level summaries of results. In general, most of the RBD nanobodies neutralized, and a large number of them neutralized quite well. So this is showing a subset that had less than 20 nanomolar IC50. What that refers to is essentially the point at which the concentration of nanobody neutralizes half of the virus in that signal. So at lower concentrations, it's unable to neutralize, and you can see here neutralization percent efficiency is quite low. As you get to higher concentrations, then of course you get complete neutralization. And so we had a range of very good neutralizers here out of a much larger subset that had quite a variable range for all the way up into the micromolar, down, as I said, into the low nanomolar range. When we step outside the RBD, we also were able to find nanobodies that neutralized. And this is an interesting finding. I believe we're still the only group to have non-RBD nanobody neutralizers identified from our repertoire. So both within the S1 non-RBD domain, and as I'll show, these are primarily within what's known as the N-terminal domain of spike. And then also quite intriguingly, some that bind not to the S1 domain at all, but to the S2 domain. And so de facto, presumably have quite different modes of neutralization versus those that are within the RBD. And as Peter alluded to, because these nanobodies are really easy to make and easy to clone, you can begin to study what the effects of oligomerizing them would be. So that's an example shown here. So if we take nanobodies that, in the case of RBD35, has a very nice neutralization profile by itself. When you add two of them, such that it's a dimer, you can see a quite strong dramatic shift, improving its neutralization ability. If you go 3 times, you can even get a little bit more. And this approach works across the board, and it even works for nanobodies where, for example, S27, this one we would have characterized as really not having any strong neutralization ability at all. Now, as soon as you dimerize it, it's now all of a sudden able to have a respectable neutralization of spike as a result. But this didn't work for everything. And so we did have examples of nanobodies that never displayed an ability to neutralize, and dimerizing them didn't, or trimerizing them didn't really offer any benefits. So as I mentioned, there's a couple of ways of testing viral neutralization. And so we wanted to test and see if our pseudovirus results paired well with some of the other assays out there. And so we collaborated with Paul Bienash's group at the Rockefeller University, and he took a subset of our nanobodies and put it through their VSV system. So this is now a VSV chimeric virus that has the spike on VSV, and performed a neutralization assay with our nanobodies. And when you look across the board, results from our lentivirus neutralization and their VSV neutralization, you can see that we actually get a pretty nice correlation, Pearson R value of 0.98. So that's quite promising. And what I also haven't shown is you can take other benchmarks, like other monoclonals and other nanobodies that have been produced against spike, and when we put them through our system, we found out that our nanobodies are performing within the same range as what you would expect from a variety of other nanobody efforts and antibody efforts. So turning from pseudovirus to live virus, we wanted to see how our nanobodies would perform in a more realistic system. So I mentioned to you that you can use these vero cells, which are quite permissible for virus replication. Colleagues of ours here at Seattle Children's had an air-liquid interface system of primary array epithelials that they use for studying asthma and a variety of other things. And so we partnered with them to see if this system would be suitable for looking at SARS infections and whether or not nanobodies would be functioning as neutralizers in this system. So this is just a little cartoon here, schematic. So you can see that these air-liquid interface cultures are pseudostratified. There's several layers to them, and you have these cilia-containing cells and then these goblets, mucus-secreting cells. And so they actually do secrete mucus, have exposed to the air, and more or less mimic what you might expect to find in our upper airway passages or the lung. And so we set up a system with them using one that they had been developing for drugs and for other sorts of approaches, both with real virus and asthma and the like, and came up with a similar system for testing SARS. So here now we're using the real SARS virus, and we do a system where we combine the nanobodies with them, and then also did experiments where we would come back and challenge after challenge with SARS, add nanobodies and see what sorts of impacts we would have on infection. The readout for this, as opposed to looking at luciferase or something like that, you're not going to be able to do that with the real virus. And so here we took advantage of another assay that we were developing at the time in the lab. This is a diagnostic assay, an RT-PCR assay that we built, a custom one, so that we wouldn't take up resources away from the global effort to test people early on in the infection. So this is a custom built diagnostic assay that we have in the lab for reading out. We've used it in clinical settings, and it's a robust assay for detecting RNA. We can detect down to our limit of detection is essentially 10 copies in the PCR tube. So it's quite sensitive for RNA detection. And just what this kind of looks like. So if you were to look top down on top of those cultures, you can see here in this confocal micrograph, I've labeled actin with this a magenta color, and you can see these essentially this nice actin stain here is where the ciliated cells are, and they kind of there's a sort of undulating pattern throughout the dish. And when you infect these and you don't have any sort of nanobody present, and then you come in and you label with spike, you can see that these cells are highly permissive for SARS-CoV-2. And in a typical culture system, we have roughly 9 to 11 million cells. And so in this system, we kind of decided to tax the nanobody. And so what we did is we set up a system where we were adding a multiplicity of infection of 0.5, meaning that we're aiming to infect half of the culture with virions, so millions of virus particles per well, and then see what would happen when we add our nanobodies on top, and if they would be able to have an effect in a system that's just being flooded with virus. And the nice thing that I'm showing here, so this is now reporting out SARS-CoV-2 copy number per nanogram of RNA extracted from these cultures, is that with the subset here, we saw really nice and strong inhibition of the ability of the virus to infect these cells and to keep those cells from being infected by addition of our nanobodies to this culture system. So the nanobodies clearly work. They're clearly great at neutralizing SARS and at blocking Spike's ability to do what it needs to do to get the virus genome inside of cells. In work, much more extensive work than I have time to present today, we went on to further structurally characterize the epitopes of these nanobodies. So this took the epitope bending data that I talked about. We also generated with the B-NASH group escape mutants for many of the nanobodies to identify specific residues where they were likely binding. We also incorporated cross-linking mass spectrometry, mass photometry, and put all of this into an integrative model to generate a structural model of where our nanobodies were binding to Spike. These two pictures here are basically summarizing the nanobody footprint from those efforts. And you can see that by and large, the RBD domain in particular has been completely saturated. We were able to find binders that are basically covering this thing on all sides. In addition to that, we were able to pinpoint for some of our S2 nanobodies their points. So this is essentially right beside the furin cleavage site, and it's actually a unique epitope because it's the nanobody is binding to subunits which come together at this point here. This is a region on the N-terminal domain where nanobodies were binding to and also have neutralizing activity. And then a little bit hidden within here, we also had N2 nanobodies that bound to this very conserved loop helix repeat region on the S2 domain that's covered by the S1 of Spike. And when you then map on how strongly these nanobodies neutralize, by and large, the strongest neutralization occurs by nanobodies that are binding within the RBD, but you're still obviously getting some neutralization for these S2 and non-RBD neutralizers. And just a quick flavor of what some of these look like. So these are now models of the nanobodies binding to their various epitopes. And we were able to then group these into classes. And so we have 8 classes within the RBD. We have 1 on the NTD and then 2 in the S2 domain. So quite a large ensemble of nanobodies. And you can see within a particular class, the way that these nanobodies bind to that region is quite variable. And this potentially has benefits in terms of choosing one over another for therapeutic development or for pairing with another nanobody in heteroligomers and the like. This is very useful information. So given that we had all of these various nanobodies and nanobody epitope classes, one experiment that seemed obvious was to see whether or not we could get multiple nanobodies binding to, for example, the same subunit of Spike. This is something that's a little bit more difficult for antibodies, monoclonal antibodies, just because of their size, but also because of the fact that they have this dimeric binding ability baked in. It's typically rare to find examples of monoclonal antibodies that are able to bind to a single subunit of Spike at the same time. And so using a method termed mass photometry, Nat was able to demonstrate that we could actually get up to 3 of our nanobodies binding to one single RBD domain at a time. So given that information, the question is that beneficial? Does that binding, having multiple binding to the same monomer, is there something about that that would impart additional benefits with respect to their neutralization abilities? We developed a series of synergy neutralization assays. And so the idea here is to test combinations, starting simply with just adding pairwise combinations of nanobodies, but of course we can build up from there and add further. And the way that this assay works is that we create a two-dimensional gradient of nanobodies on 296-well plates. So you can see nanobody 1 and 2, and they're going from high concentration to low concentration in one dimension, and the other nanobody does the same in the other dimension. We can then obviously have each nanobody by itself, as well as positive, negative controls. And all of this then can be fit onto 384-well plate and read out using the neutralization assay I spoke about. And for this, I mean, we used automation quite extensively throughout for our neutralization assays, as well as the RT-PCR assays. But the advantage of having liquid handling robots in the lab really allowed us to reliably prepare these two-dimensional plates consistently and rather rapidly, and really was a benefit for the subsequent analysis that I'm about to demonstrate. So I'll just take a step aside. I know that there had been some interest in automation. Our lab, we have several liquid handling robots. Everything is pretty on the smaller scale of automation, I would say, and it's great for assay development and for complementing the sort of molecular biology work that would go on within most academic labs. Obviously, within industry settings, you can really scale this up and make this a lot more specialized for a specific workflow, but the flexibility of having these small scale liquid handlers is great for the sorts of experiments that we've been generating here to characterize these nanobodies. Just as a quick visual demonstration, so here's the 2D plate, and now we're adding it to that 384-well plate here. And we're able to, with this specific liquid handler, get very precise. The error in pipetting on this particular liquid handler is quite low. It's less than 0.5% of pipetted volume. So it's much better than manual pipetting for sure. And the ability to be consistent in pipette steps and mixing steps really reduces variability in your assays as a result. Plus experimental error, pipetting. I'm sure many of you who do these assays by hand, it's not fun pipetting into a 384-well plate, even when you do have multi-channel pipettes. So that's one advantage. Another advantage I thought I would show here quickly is just to demonstrate when we set this up. So when you pipette into small channels and you're dispensing cells, for example, you really have no control over the ability of these cells to dispense themselves evenly within a plate. So this is a manual dispense. It's not a bad job, but you can see that as a result, most of the cells are clustering around this half of the plate and are pretty sparse up here. Whereas with the liquid handler, you get a much more even dispensing of those cells across the plate. And as a result, this will grow into a nice even monolayer. And we know that from well to well, the variability in that monolayer is going to be relatively small. Whereas with the manual, you're going to have large regions potentially. You'll have certainly cells or wells that will look like this, but you're going to also have many wells that look like this, or they're going to be, this side of the plate is going to be absent of cells. And when you then begin to add your virus particles and the like, their ability to interact and uniformly infect this monolayer is impacted by that variability. So that's one advantage of liquid handling. So returning back to the assay itself, once we've done this and we've added them to cells, we then generate these two-dimensional luminescence maps. And so what you're looking at here, this is a region of low nanobody concentration for both nanobody 1 and 2. And so then it has it ends up lighting up because the virus is getting in and producing luciferase, whereas this area here is relatively dark, and that's because in these concentrations, the virus is being neutralized. So how do we interpret this, and how do we derive synergy, whether synergy is happening and what kinds of synergy is happening from something like this? And to do that, let me illustrate this with a case where we chose two nanobodies that are binding the same epitope on spike. So this is S127 and S123. If you look at their IC50s, they have very respectable and very similar IC50s. And so the idea of this here is that when you were to add them in a pairwise combination, like I demonstrated before, you would naturally expect that essentially along the plane at which you have 1-to-1, in which all you're doing is really just doubling your nanobody concentration. And so when you calculate out what you would get from just an additive effect, the IC50 should go from 3.3, 3.7 combined down to 1.7. And we see that our measured IC50 1.3 is within within error of 1.7. And this is the sort of thing that you would expect for nanobodies that do not synergize. So everything's working there. Now, if we move over to a situation where we do have the ability of both nanobodies to bind to different parts of RBD at the same time, that's with S123 and S11. You can now see that sort of hockey stick shaped neutralization curve that I demonstrated before has now got this little wobble in it here. And these have slightly different IC50s. When we calculate what you would just have by additive, you get something that's 1.2, but now you can see that the measured IC50 for this is much greater. It's actually 0.46. So this is just a simplistic way of demonstrating this. In reality, what you need is a little more complicated modeling methods, and there's a variety of open source packages out there that are accessible for looking at drug combinations and whether they synergize or don't synergize. And we took advantage of several of them and generated a series of performed a series of studies with our nanobodies to test and see if they neutralize. So this is the same curve as I just showed in the previous slide. And then what we've done is we've looked at what's the difference between this curve here and what you would expect if these nanobodies did not interact and had just that sort of regular hockey shape. And the difference between those two models is what we call then a synergy plot. And you can see then that within these concentrations, these nanobodies are enhancing the synergy, the neutralization of each other quite dramatically. What I put on the right here is where I've coded on spike where these nanobodies bind. And so you can see they're binding on either side of the RBD. And so simultaneous binding to either side of the RBD leads to, in this case, it ends up being about a 20 to 30-fold improvement at this concentration for these two nanobodies. And to make a long story short, we've looked at a variety of different situations. If you look outside, so if you take an RBD and a non-RBD, like the NTD region or the S2 region, these RBD nanobodies end up really enhancing the neutralization that was occurring both for the NTD and for the S2, sometimes up to several thousand-fold for those nanobodies in combination. We also had an example, a really interesting one, where in this particular example, this S146 is binding to a region on the spike that actually prevents, once it binds, it prevents binding of the other nanobodies. So it kind of locks spike in a configuration that prevents binding. And so you actually see very modest synergy, and that's because it's actually antagonizing the other nanobody's ability to neutralize. So that was also quite interesting as a result of this. Okay, so that's all fine and good, but of course, as we've seen within the past year, SARS is evolving and continues to evolve, and with the latest variants has introduced a number of point mutations that have gone across particularly receptor binding domain. This has then led to most of the monoclonal therapies, antibody therapies that have been out there for clinical use to be called back because they no longer function. And most of the nanobodies that have been published also no longer function as well. There had been a preclinical nanobody in development as a therapeutic, but that one had been rendered obsolete by Omicron. So question that we had was, to what extent does the repertoire, we've surface coded this structure, to what extent does the repertoire hold up against such a thing? And to do this, we've been using both the pseudovirus assays, but also testing the actual variants of SARS. This is an example of a plaque assay here, where we have one nanobody that's able to still neutralize. So it's preventing plaques forming at these higher concentrations. And then at lower concentrations, the virus gets in. Whereas this one, which was a pretty good one for a while, this one no longer is sensitive against that variant. So it's a very real problem. And it's something that we obviously are all concerned about with respect to not only developing therapeutics, but also the rate at which vaccines, our own monoclonal response, antibody response to from vaccines, how they'll stand up against these evolving variants. And so our current work really has been to characterize this out, and I'll just highlight some of our unpublished but soon to be published data, looking across the repertoire now, both at Omicron and Delta in neutralization assays. And we see that, yes, it's true, there is a number of classes where Omicron and Delta and Omicron-related variants render them obsolete. But for by and large, many of them, we still actually have retained quite good neutralization. And this is a little bit surprising, the fact that we still had some groups that are quite good. And I think it comes down again to the small size of the nanobody and the ability of the nanobody to really engage epitopes on spike that are not going to be under immune pressure from our antibodies, from the much larger antibodies. At least that's the interpretation that we currently have for this data set. So just to wrap up here, before the Q&A, we're currently testing our nanobodies. We have animal models in the lab, looking at both prophylactic and therapeutic use cases for those. But we're really interested, if anyone on the call is excited by this work, and we would love to partner with people both in therapeutic development, but also with diagnostic potential. I think there's a phenomenal opportunity to be had here with these nanobodies. We've really only just begun, as I said, to sample the repertoire in both in terms of there's a much larger repertoire out there, but also the combinations that can be put together, and the synergies that we see that you really don't get with monoclonals because of their large size. Their inability to pack so many onto a subunit is one disadvantage there, and one real strong advantage for the nanobodies. As well as, we still also are generating variant of concern specific spike nanobodies and nanobodies to other targets on SARS as well. So just to summarize, I think the take home from what I presented today is that there really is strength in numbers. You want to have a large repertoire of nanobodies. I think this approach that we've demonstrated with SARS can be rolled out for, and we're looking at that for other viruses, for other antigens of interest. You want to have a large repertoire. You want it to be a quality repertoire. So the biophysical characterization showing the strong binding is key. The neutralization assays are largely reproducible. Pseudovirus, real virus, we've tested them all, and they all seem to be great ways at testing nanobodies in these virus assays. Automation has really been helpful in assisting in our development, as well as in the readout and reproducibility and the throughput. I think the really cool thing is that even with Omicron and the new versions of Omicron that are coming out, the nanobodies are still very potent against them. And I think in the future, we want to look at combining nanobodies either as cocktails or as heteroligomer formulations to really take advantage of this synergism that we discovered using this platform. And just finally, I'll just give a quick plug. So the NCDIR is a place where you can go and check out some of the resources that we have with respect to nanobodies and with respect to a variety of other technologies. And so feel free to check that out. We do a lot more. There's sample preservation methods, automated affinity capture, mass spec methods, structural modeling, and of course the nanobody work that we have. But at this point, I'll be happy to take any questions.
All right, thank you, Fred. So if anyone has, we've answered a lot of questions in the chat already, but if there's anything additional for Fred, please put your question in the Q&A section. For starters, though, a couple questions about animal challenge models. So have you tried the nanobodies in animal challenge models? SARS-CoV-2 monoclonals with a limited FC function have had compromised therapeutic efficiencies. So it'll be interesting to see how nanobodies perform. And it's kind of a related question, how the FC domain matters here.
Yeah, these are great questions. So nanobodies, don't have an FC, so they will not function based off of effector function. But they could, I mean, we can add them. Other groups have done this. They have added FCs and have shown in rodent models that those nanobodies are capable of helping animals recover from an infection, both in therapeutic formulations and in prophylactic. We're currently using just the nanobodies themselves. This is really something that's was quickly bypassed, I think, by the community. Adding the FC seems to be the industry standard, so a lot of people are doing that. And we've been doing mouse models right now, currently are ongoing, both with nanobodies alone, but also in combination. And testing their ability to protect mice from infection. And so I'm preliminarily, this looks really good, but this is ongoing work.
So there are also some raised questions if anyone wants to ask their question live. I'm not sure I can enable you to speak, but. In the meantime, what about accessory proteins as epitopes? Have we thought about that? Actually,
Sorry, say that again, Peter. What was the question?
Accessory proteins as epitopes. So other proteins than spike, I think is the question.
Other proteins than spike. Yeah. So spike is the dominant surface glycoprotein. Some of the other surface proteins like envelope, membrane are a little more challenging because of their they're quite small. Their surface that's exposed on this to the cytosol or to the nanobody is quite limited because they're primarily buried within the membrane. But theoretically, it's a potential target. We do have nanobodies against nucleocapsid. We haven't tested them in terms of their ability to function as a therapeutic, but you could think about delivering nanobodies using other methods to get them into cells, in an infected individual where that might be applicable. But we haven't really explored that.
So if we can get Prem to unmute, they can ask their question. Hopefully they're enabled. If not, could you say more about these softwares that are involved in the synergy modeling?
Yeah, certainly. So rather than reinvent the wheel, we've taken advantage. There's a number of open source softwares that are found both for the common, most people like to use the R package for scientific, computing, biostatistical analysis and the like. There's one model there that's called the braid model, which is really good. And we've used in this modeling work. The other one that I really like is a Python package called Synergy. And so if you just search Python synergy, you'll get it. It's a really nice package. They have a variety of synergy models, including their own, but you can compare these models and within the same package, which is quite handy.
Okay, another question from the chat or from the Q&A. Does priming and boosting with spike antigen improve the breadth and quality against other variants or other related coronaviruses?
Yeah, I think the answer is yes. We have evidence of that. Certainly, with the camels, with the llamas, the response from a prime and boost with the variants has led to a large response that I'm sure helped existing circulating nanobodies, as well as generating new variant-specific nanobodies as a result. I think I did a little bit of a poor job of explaining that, Peter, if you want to provide a little more color on that one.
I think you covered it. We're certainly looking into it. We kind of early days on that front. We'll try one more time if someone can raise their hand. So ZZ, if you can raise your hand, and we might be able to enable you to ask your question. So maybe not. If you have a question, I think best to just put it in the Q&A, and we can read it for you.
Peter, did you answer Ellen's question about screening nanobody library?
Yeah, just to briefly explain. So rather than phage display, the screening approach is purifying antibodies from sera, and then by mass spec we can just pick the ones that are specific for the antigen. We pull out any antibodies that bind that antigen, and then we can use mass spec to get the identities of those candidates to then correlate with a separately constructed sequence library. Another question is about how long the immunization lasts. So I can quickly answer that. We normally do about a 3-month standard immunization cycle, 4 boosts over 3 months. But we find that particularly these heavy chain only antibody variants, they will just continue to be enriched at an even longer time scale. So coming back after an additional 6 months, even a year, you just kind of get more and more of a response. So sometimes that extra time can be very helpful.
Yeah, and spike was particularly immunogenic, right, Peter?
Yes.
It gave a really nice strong response.
Yes. It definitely it makes a big difference, how nicotogenic your protein is, and particularly with with infectious disease targets, that can be very much the case. To answer one of the questions, the recording will be available on the website later, I believe. And then one more from the Q&A. So are you able to humanize nanobodies or convert them to human monoclonals?
Right, great. This is a great question. So yes, there are amino acid substitutions within the framework region of the nanobody that can be undertaken. We've done this with a bunch of ours. Pretty much everything that we've brought into the animal model, we've first gone through a step of quote unquote humanizing them and verifying that they still function after these amino acid substitutions, and they do, and it likely will help reduce the immunogenicity. But nanobodies, aren't very immunogenic to begin with. They're rather small, and so it's available. You can humanize, but they're also not very immunogenic to begin with.
Yeah, I mean, various studies have looked at this and found that essentially humanized versus unhumanized in practice, your odds of having an immune reaction that's problematic for a therapeutic is kind of the same in either case. But it is still common practice just to be sure. One last question about whether we use adjuvants. I quickly can say, yes, we do. We use complete Freund's adjuvant typically for the initial boosts, followed by incomplete for the subsequent boosts. There are other options out there, though, that work pretty well. Another related question is, have we considered the ADE effect, the antibody-dependent enhancement?
Yeah, so I think that's really where nanobodies shine. So antibody-dependent enhancement, for those who don't know, is a situation that arises where you have an antibody binding to a virus, but it's not neutralizing the virus. So this typically happens where, for example, in dengue, there's 4 distinct serotypes that circulate of dengue out there, and antibody responses to one don't neutralize the other because of mutations in the E protein. What ends up happening is that they still bind, don't neutralize, and then any FC receptor containing cell can bind to that antibody, and you essentially get an enhanced infection. It's a new way for the virus to get in. It also gets into typically immune cells, and you can get a very robust and strong viral infection as a result of this. So, and this was initially a concern with SARS-CoV-2, although the evidence that this happens is pretty rare. So typically, we don't worry about antibody-dependent enhancement with SARS-CoV-2, although SARS-CoV-1, I think it does happen. The nanobodies don't have an FC region, and so when either they work and neutralize, but if they bind and don't neutralize, there's no receptor for them. And so we really avoid antibody-dependent enhancement when we choose a nanobody as a therapeutic.
And going back to the in vivo use here, so what kind of half-life do we see, and is there a way to make them more stable in circulation?
Yeah, sure. So without the large FC region, the nanobodies are pretty short in their half-lives. So in a mouse lung, I think that's, we see it sitting around maybe an hour or 2 or something like that, and then just by itself, and then it would go away. And so if you want it to stick around longer, you need to use a strategy to retain it. And so most people add an FC for that reason. They'll add an FC region to nanobodies, or you can add nanobodies that are able to bind to albumin, for example. So albumin binding nanobodies, dimerizing with the SARS-CoV-2 spike nanobody improves the half-life of the nanobody. But it's really an advantage in the ability to select which strategy you want to tune that half-life. And in the cases of, certainly in the cases of certain pediatric treatments, the ability to have a quick half-life, specifically if you're going to nebulize something into the lungs, is beneficial. So we can have a rapid turnover, or we can prolong it by these other methods.
All right, great. I think that's all the questions we have right now.
Okay, great. I think then we'll wrap up here. Thank you everyone for sticking around until the end. Thank you so much, Fred, for this amazing talk. Really great work. And for the lively Q&A. Also, thank you so much, Peter and Natalia, for answering questions in the chat and making this a really lively webinar. If there are any more questions, please go to our website. We have a Q&A section there. You can keep posting questions. As you'll see, there are already many questions, and Fred and Peter have already answered several of them. But keep asking questions. We will keep inviting experts to answer those questions, because we really want to start building a community here where you can all keep on discussing today's webinar topic. As mentioned before, this webinar was recorded and it will be posted hopefully in about a week or so. We will send an email when it's available. And we have another webinar coming up on November 13. The title is Label Retention Expansion Microscopy with iodine denoising. There's a novel denoising tool developed at the Chan Zuckerberg BioHub in San Francisco. And so we'll have 2 expert speakers talking about that in just about 3 weeks. And for any other information regarding future webinars, please visit our website. We have many more webinars coming up. And then thank you again for staying, and hopefully see you next time. Bye-bye.